Hey, what's up? It's George. Welcome to our biology project. In case you didn't know already, it's about cellular respiration. Your body's got these things. They're cells. Neat, huh? Cells do fun stuff like keep you alive. In order to do the fun stuff, though, your cells need energy. Have you ever eaten anything? Probably. When you eat things, you get all kinds of reactions from your cells as they process it, and make energy for your body to do stuff. Pretty simple, right? If we were to summarize it quickly though, your cell breath is split into a few parts that goes something a little like this. At first you go through a process called glycolysis, which takes place in the cytosol. Then you have your mitochondria matrix. It's where all the fun stuff happens. The process of pyruvate oxidation occurs, which uses the stuff produced by glycolysis and prepares it for entry and use in the mitochondria. Once it's been prepped, it gets circulated through the citric acid cycle, or Krebs cycle, whichever you prefer. And while this is all happening, there's some NADH being sauced around along from the ride from glycolysis and your boy Krebs. It's not until the final step of electron transport that some flavin adenine dinucleotide, or FADH2, that was also produced by Krebs is used as well. So what's so important about all this jazz? You probably already knew, but adenosine triphosphate, or ATP, is the name of the game here in Cellville, and cellular respiration is the talk of the town, because it's a relatively easy way for you, as a human being, to get some shiny ATP for all your cellular needs. So to begin, we may as well start in chronological order, right? That means we're going to talk about this green guy, glycolysis. Glycolysis is convenient in that it can be summarized into two phases, the investment phase and the payoff phase. Similar to how you can invest in cryptocurrencies, but in this case you won't lose all your money. Ah yes, the investment phase. When you consume food of any nature, your body breaks it down into its simplest molecules so that they can be used as necessary. In this case, glucose molecules are being absorbed, and the process of cellular respiration can begin. The investment phase consists of quite literally investing ATP for the greater good of producing more. During the first step, ATP is manipulated by the enzyme hexokinase, which donates a phosphate group that attaches to the glucose molecule, producing glucose 6-phosphate. The next step consists of phosphoglucosamidase, rearranging glucose 6-phosphate into fructose 6-biphosphate through isomerization. Then, a second phosphate group, derived from ATP, is attached to fructose 6-biphosphate, rendering it fructose 1,6-biphosphate. Furthermore, fructose 1,6-biphosphate is then split into glyceraldehyde 3-phosphate, also known as G3P. Alongside dihydroxyacetone phosphate, known as DHAP, finally, this DHAP is converted to G3P as well, producing 2 G3P per glucose. We're not done yet, of course. Now we have what we've been waiting for, the payoff phase. This phase is split into another 5 steps, with each occurring twice. Firstly, triose phosphate dehydrogenase is used to remove 2 electrons and 2 hydrogens from G3P. Some of the energy released during this process is trapped by adding an inorganic phosphate derived from the cytosol. The removed electrons are accepted by NAD+, along with 1 hydrogen, making NADH, while the other hydrogen is released into the cytosol. This entire process leaves us with our new friend, 1,3-biphosphoglycerate. The next step involves the use of phosphoglycerate kinase, which transfers one of the 2 inorganic phosphate groups to ADP to produce ATP. The moment we've all been waiting for. Except not quite. We're left with 3-phosphoglycerate, which means we're not done yet. Now phosphoglucomutase gets involved again and rearranges 3-phosphoglycerate to make 2-phosphoglycerate. Then, with enolase, electrons are moved from one part of 2-phosphoglycerate to another, resulting in phosphoenolpyruvate and a loss of water through redox reactions. Finally, the remaining phosphate group is removed from phosphoenolpyruvate to transfer to ADP, which forms ATP and pyruvate. And just like that, you're all set for pyruvate oxidation. And on top of that, you've not only started the process to making a ton of ATP by sending pyruvate to the citric acid cycle, you've also received some of your initial investment back. Now that's value. And of course, during this whole spectacle, 2 NADH is being kept for later as well, alongside some water waste. Now that we've finished learning about the basics of investing and getting our money's worth, what do you say we take a look at the next big step of cellular respiration, the citric acid cycle? This cycle is commonly referred to as the Krebs cycle, which is conveniently named after the guy that discovered it. Now, as mentioned before, this is where all the fun stuff happens. Once glycolysis has finished having a ball with the glucose extracted from food, it becomes this somewhat beautiful gift-wrapped package that can now be sent on its merry way into the Krebs cycle. Well, almost at least. Right before it can be sent on its merry way, there's one last step, which involves the process known as pyruvate oxidation. Once pyruvate makes it to the mitochondria via transport proteins, they're stripped of all their precious carbon dioxide. It's actually just a waste product. Once this has occurred, they receive the all-clear signal from everyone, and into the citric acid cycle they go. The cycle begins and continues to cycle through this point, where an 2C-acetyl group carried by CoA is transferred to oxaloacetate, forming citrate. Then, citrate is rearranged by the enzyme aconitase to isomer isocitrate. This isocitrate is then oxidized to alpha-ketoglutarate. During this process, one carbon is removed and released as CO2, and our friendly NAD+ is reduced to NADH plus H. The release of CoA from succinyl-CoA produces some stuff known as succinate. Also, the only ATP actually produced during the citric acid cycle is produced here, when GDP is converted to GTP, which causes ADP to become ATP via substrate-level phosphorylation. What a mouthful. But don't worry, we're not selling you short. There are still steps to finishing up the Krebs cycle. Our shiny new succinate can be oxidized to become an even fancier molecule, fumarate. During this conversion, 2 protons and electrons are removed and transferred to that FAD we had sitting around left over from earlier, producing FADH2. Our fumarate is then converted even further to malate by adding some good old H2O. Finally, the malate is oxidized for a final time to become oxaloacetate and reduce NAD+ to NADH plus H for a second time. This final product can react with acetyl-CoA to re-enter the cycle if it so chooses. I mean, freedom of choice is always welcome, right? So in summary, we've sent 8 NADH and 2 FADH2 as somewhat of an investment package that'll give us a ton of ATP in the end, towards the next step of cellular respiration, the electron transport chain and chemiosmosis. Of course, we've also produced a total of 4 ATP along the way. We're already making profit. Now what do you say we make even more? Electron transport and chemiosmosis would like to have a word with you and how you can make and own all the ATP you've ever wanted. Because oh man, all that hard work and long list of processes are finally paying off. So our final journey officially begins with the exit from the Krebs cycle into the electron transport chain. Occurring in the inner mitochondrial membrane, this process is all about transferring electrons from NADH and FADH to oxygen. It can be easily summarized in 4 complexes. The first being complex 1, NADH dehydrogenase, complex 2, succinate dehydrogenase, complex 3, the cytochrome complex, and complex 4, cytochrome oxidase. That's a lot to remember, sure, but in practice, it's not so complex as the complexes would have us believe. In essence, a shuttle known as ubiquinone moves electrons from complexes 1 and 2 to complex 3, and a second shuttle known as cytochrome C moves electrons from complex 3 to complex 4. And what's the importance of all this electron movement? Basically, we're trying to set everything up in the party room of complex 4, where all the super electronegative oxygen we breathe in visits to pull all the collected electrons away, and allows the transport chain to continue its transport chainness. Now you're probably thinking, this is an awful lot of spectacle, but what's the end result? Where's my ATP I've clearly worked so hard for? Well, my friend, if patience wasn't so easily tested, it would hardly be a virtue now, would it? We have one final step to retrieving all of our hard-earned ATP, chemiosmosis. We won't delve deep into the specifics, but chemiosmosis is a process in which ATP is synthesized using energy produced from an electrochemical gradient. Remember that oxygen that was taking all the electrons away from complex 4? Well, some hydrogen ions really were dying to meet the fabled oxygen and got trapped in the mitochondrial chamber during their excitement. This creates an imbalance between one side of the mitochondrial membrane and the other, and creates a driving force to move the hydrogen ions around. What I'm trying to say is that chemiosmosis is actually the use of this driving force so that these hydrogen ions can get where they need to be going, the ATP synthase. Yes, the moment we've truly all been waiting for. This synthase acts as a motor that opens and closes channels so that trapped hydrogen ions can pass through freely. It possesses a really neat headpiece that does what we've wanted all along, combine ADP and an inorganic phosphate to make ATP. So with all that said and done, we've finally unpacked our packed 10 NADH and 2 FADH2 that was painstakingly produced during this whole lot, alongside our initial 4 ATP to make a breathtaking total of around 32 to 34 ATP, but your mileage may vary. And that, my friends, is the story of how you too can be a proud proprietor of ATP. Hope you enjoyed.